who is part of the team of experts who discovered the Higgs boson particle, theorized by the 2013 Nobel Prize winners Francois Englert and Herd Higgs. So please join me in welcoming President Cueto.
Thank you so much. What a wonderful crowd. Welcome. This is fabulous. No, it really is. It's so great when you see a packed room, standing audience only. So, although the people standing, I'm sure, are not that comfortable, but so good afternoon and welcome to the 8th annual Sharon Cosloy Edward Blank Lecture at City College. Ed Blank established the lecture series in memory of his late wife, Professor Sharon Cosloy. Professor Cosloy was a member of the City College biology faculty for 27 years and served as the chair of the biology department for 6 years. I actually, even though I've only been here 3 years, I had the privilege of knowing her because Sharon shared a party wall with my husband's laboratory, and my husband, Richard Cueto, was on the tenured faculty here for over 20 years. She was a wonderful teacher, a beloved faculty member, and a superb researcher, a mentor, a wife, mother, and the most passionate investigator at life sciences that I've known. I'd like to thank Ed. I'd like to thank you, Ed, and your family for this wonderful lecture in memory of your wife. After her death in 2001, Ed Blank made successive commitments to establish a scholarship in her memory. The Sharon Cosloy Professional Fund and the Sharon Cosloy and Ed Blank Lectureship in Biology. Will the Blank family who's here please rise so that we can give you an applause? That would be Ed, I don't know if Rebecca and Michael, is Michael here too? Thank you all for allowing our students, faculty, and community to be exposed to such a wonderful, wonderful resource and opportunity. I also want to acknowledge our Dean, Tony Liss, who could not be here. He's out of town tonight, but he really expresses his regrets and sends his best wishes for, again, a wonderful, successful lecture. And I also want to extend a special thanks to Chris Lee, because Tony's relatively new here, and Chris was the interim dean for another year, and she's been tremendously instrumental. So thanks to you, Chris. And finally, I wish to recognize the Cosloy professor, who is Anudrada Janakaraman. Who's Anu? Anu. There you go. I'm looking around and saying, Anu, please, Anu. How do you pronounce your name? Janakaraman. Anu Janakaraman. Okay. I also want to highlight our two scholarship recipients, the two Cosloy Student Scholarship, 2014. Andrea Cardenas, where are you? Please stand. And Maisum Begum, please stand. Please stand. And I'm going to read the following about each of you. So if you will each stand while I read the following. Come on. Andrea and, right, Maisum. Okay. Andrea attended high school in Ecuador, where she also attended 1 year of medical school university before coming to the United States. She plans to go on to graduate school and medical school to become a pediatrician. Her project is the interaction of CLP-XP and SAPC and E. coli cells in Dr. Anu's lab. So, thank you. Okay. And Maisum Begum attended Francis Lewis High School in Queens. He started studying law, but realized in his senior year of high school that he wanted to do science when he took a college-level course in the sciences. He also plans to go to medical school, but wants to take a gap year, possibly to study abroad in Japan. He looks forward to testing all the various types of medicines during the first year in his medical school to find his perfect fit in field. His special project is identifying the role of APL1 in Dr. Chris Lee's lab. So congratulations to both. And now, on to the event of the evening. It gives me great pleasure to introduce our guest lecturer, Dr. Joan Stites. She will present a lecture entitled, as you see, Non-Coding RNAs with a Viral Twist. Our understanding and appreciation of RNA, ribonucleic acid, as a central player in how genetic information is processed, owes much of its progress to the work of Joan Stites. She and her colleagues are responsible for a breakthrough in understanding how RNA is spliced. No introduction of Joan would be complete without mentioning her work on behalf of women in science. She is a role model. You really are a role model for women everywhere. And I'm a woman in science, and I have always viewed you as a role model for me. You lead by example, you mentor, and you train a large number of women scientists. And for that, those of us who are women in this audience are very grateful to you. Thank you. Professor Stites earned her PhD from Harvard University. She continued her scientific career in the famous laboratory of molecular biology at the Medical Research Council in Cambridge, England. In 1970, she joined the faculty of Yale University, where she is currently Sterling Professor of Molecular Biophysics and Biochemistry. She is also a Howard Hughes investigator. Additionally, Joan Stites is a member of the National Academy of Sciences, the Institute of Medicine, and the American Academy of Arts and Sciences. She is also the recipient of many awards, including the National Medal of Science, the Gardner Foundation International Award, the Albany Medical Center Prize in Medicine and Biomedical Research, and the Weitzman Women in Science Award, and the L'Oreal for Women in Science. Please join me in welcoming a dynamic, incredibly intellectual speaker and scientist, Dr. Joan Stites.
I have to start out by saying, wow, this audience portends great things for the future of RNA research. And I hope you will all stick with it. This is wonderful. But I also need to say thank you to the Blank family, and thank you, President, for those wonderful words. And I've had such a great day. I've talked to old scientific friends, new scientific friends and colleagues, as well as a wonderful group of students at lunch. And I've just had a wonderful day, and it's so nice to be here. Thank you very much. So, as you've already heard, and as you see, I'm going to be talking about RNA, specifically about non-coding RNAs. And just as our genomes, the cellular genomes, encode non-coding RNAs, so do some viral genomes. Not all viral genomes, but some viral genomes, and I'll explain a little bit more about that. And as with cellular non-coding RNAs, the question is, what is the function of these RNAs inside cells? How are they contributing to the process of gene expression or other things that they might be doing? And there are a couple, so with viruses, you would think that either they might be doing something with respect to enhancing the viral life cycle, or they might be helping to defend the viruses against the offensive tactics that the host sets up once it gets infected by a virus. There are a couple of things to remember about viral non-coding RNAs before we begin. The first thing is that because viruses have limited genomes, if they're going to go to the bother of making a non-coding RNA, it ought to be doing something pretty important for the virus, either, as I mentioned, in the life cycle or defending it against the host offense. And secondly, viruses don't make things up. They acquire pieces from their host cell. But just because you find that a virus is making something that looks like something that you know about in the host cell doesn't mean it's doing the same things. Viruses pick these things up and then very cleverly manipulate them so that they can contribute to the viral strategy in the end. And finally, as with all studies of viruses, we learn about evolution, and we learn about the evolution of the host and of the virus simultaneously when we study viruses. So I'd like to introduce the subject of non-coding RNAs with this wonderful slide that comes from the bioinformaticist John Maddock in Australia. And what he's plotting here is the amount of non-coding DNA in the genome versus a variety of organisms that are aligned here as a function of developmental complexity. Now, this is sort of a mystical term, but I think you all sort of get a feel for what this might mean. And so what we're looking at here is all these blue guys down here are single-cell bacteria. And they have usually 15% or less non-coding DNA. The black organisms here are single-celled eukaryotes like yeast and fungi, and you see the amount of non-coding DNA rises. Here we have plants and invertebrates, and when we get up to vertebrates and mammals and humans, we have a staggering 98.5% non-coding DNA in our genomes. And one of the truly remarkable things that's been learned in about the last decade is that probably at some time, virtually all of this non-coding DNA is transcribed into RNA. So even if these RNA molecules might be very short-lived or very low in abundance, it's still possible that they're doing important things in cells, like contributing to signaling or regulation. And we do know, because of the studies of the last several decades, and you already heard a little bit about this in the introduction, that there are a number of ways that non-coding RNAs contribute to the regulation of gene expression. So let me just go through this a little bit. You heard mentioned the snRNAs, the small RNA molecules that with their associated proteins recognize the ends of introns and assemble into a spliceosome with a lot of other proteins to remove introns from pre-messenger RNAs. It turns out that there are non-coding RNAs that are involved in various aspects of transcription, like 7sk RNA. In the nucleolus, there are so-called SNO RNAs, small nucleolar RNAs, that are there to put very specific modifications into ribosomal RNA that makes it function better. In germ cells, there are pi RNAs that are helpful in keeping transposons from moving around and wracking havoc with the genomes in germ cells. And finally, there's an ever-growing, there's a new paper every day set of large non-coding RNAs. One of the best known examples exists that interacts with chromatin and therefore serves to regulate on the transcriptional level whole sets of genes. And when we go out to the cytoplasm, there's the experimentalist tool, namely you arrange to make a small RNA inside cells that's perfectly complementary to a messenger RNA you want to target for destruction, and that gets together with the AGO and other associated proteins, and then you get cleavage of that RNA. And of course, the machinery for this is derived from this whole new class, one of the latest large classes of non-coding RNAs, the so-called microRNAs. So these are 22 nucleotide RNAs that pair imprecisely with a messenger RNA, and therefore, instead of directing cleavage, direct translational repression, or in some cases stimulation or shortening of the poly-A tail, and therefore the level of the messenger RNA. And of course, in the cytoplasm, we have the classical non-coding RNAs, ribosomal RNA and tRNA, and the roles that they play in translation. Now, this is only the tip of the iceberg, only shows you some of this, and it's clear that there's lots more to be discovered. The other thing I want to point out is that all of these non-coding RNAs carry out their function associated with tightly bound proteins. So they function as RNPs, ribonucleoprotein particles. So the non-coding RNAs that we've been studying that come from viruses all come from a class of viruses called gamma herpes viruses, and these viruses infect primate cells. And what you're looking at here is the name of the non-coding RNA or RNP. The virus it comes from, you probably know these two, but I'll tell you also about this one. And then over here, how we're doing with respect to finding functions. So let me make a couple of general comments. So gamma herpes viruses have large double-stranded DNA genomes, so they encode like 100 to 200 genes. They are all lymphotropic viruses, so they infect either T cells or B cells. They're all oncogenic viruses, so they're associated with cancer formation. And finally, a very important thing about all of them is that they have both lytic and latent states. So they can go into a cell and replicate and produce more virus, or very often they'll go into the cell, their genome will be there, it will be producing a few viral gene products, but they basically won't be doing anything except getting carried along as the host cell replicates. And what you see by comparing, these are the dates of discovery of these non-coding RNAs, some of them going way back. And if you compare those to the number of question marks we still have over here, you can see that finding functions for what these things are doing has been difficult, very challenging, the most challenging problems I think my lab has worked on over the decades. But recently, we've been making real progress and coming up with molecular explanations. And I think what's happened is that technology has finally caught up. And now we're able to ask questions and do things in a way that we just couldn't before that actually help us illuminate what's really going on. So obviously, I'm not going to try to tell you about all of these. I'm going to tell you two stories, one about one of the RNAs in this class, and I'm going to start with telling you about a larger non-coding RNA that's in this particular class. So I want to start by introducing you a little bit more to KSHB, Kaposi sarcoma-associated herpes virus. And this is associated with cancers and other lymphoproliferative disorders in people that are already immunocompromised, like people who are suffering from AIDS. And it's the causative agent of these ugly sarcomas that you see over here. And the RNA we're going to be talking about, the non-coding RNA, is made during the lytic phase, so when the virus is replicating. And it's called PAN for polyadenylated nuclear RNA. And this RNA resembles a messenger RNA in many respects. It's transcribed by RNA polymerase II, therefore it's 7-methyl-G capped in the normal way. It has a poly-A tail that's made in the normal way by recognizing a signal, cleaving downstream and adding a poly-A tail. However, this RNA appears to be pretty exclusively nuclear. It certainly isn't exported in bulk to the cytoplasm, and there aren't any really long open reading frames, although I must confess that another lab is about to publish a paper saying that it does give rise to some short polypeptides that you can see encoded, and maybe these are RNAs that get out during mitosis, so they come in contact with the protein synthesis machinery in the cytoplasm. The really remarkable thing about this RNA is it accumulates to about 500,000 copies in the nucleus of a lytically infected cell and can account for up to 80% of the polyadenylated RNA in the cell, which is a huge amount. So this raises a couple of questions. Obviously, what is this RNA doing for the virus? And secondly, how does the RNA manage to accumulate to these very, very high levels? We know a little bit about the first question, and I'll just summarize those results in the next slide. And what I'll really be telling you about is what we've learned about RNA structure by studying this RNA that explains how it's resistant to degradation by nucleases and how it can rise to such high levels. So let me go along with another introductory slide. Okay, what's responsible for this RNA accumulating is an element in the RNA that's near the 3' end. So I said this whole thing is about a kb long with the typically length poly-A tail. And this element is called ENE, and there's its sequence. It's 79 nucleotide elements necessary for It has to be there in order for the RNA to accumulate to high levels. But the other thing that the RNA has to have is it has to have a poly-A tail. And you'll see why this is in a moment. And these basic discoveries were made by Nick Conrad when he was a postdoc in the lab. He now has his own lab at UT Southwestern. And the way the E&E acts is to counteract a very rapid nuclear decay pathway that acts on intronless polyadenylated RNA. So there are no introns here, but is polyadenylated RNA. And you can actually take the E&E element and put it into another RNA that doesn't have introns and is polyadenylated, and it will help that RNA accumulate to high levels. What Sumit Bora, who's a recent graduate student in the lab, did in his thesis was to show that pan-RNA is important for the expression of late viral proteins. So that's its function in a general sense. And it's therefore necessary for the production of new infectious virus. But we don't know the molecular details of how that occurs, so I'm not going to talk about it anymore. And that work then was followed up by a comparable conclusion from another lab. The question I am going to tell you about is the question of how does this E&E, how does this element work to protect this RNA from very rapid degradation? And by the time that Conrad left the lab, he had done a lot of biochemical experiments and mutagenic experiments, and we thought we had a model that we were pretty sure was going to turn out to be the right model. It had to be right, it just fit everything. And the idea was, this is the way this element would fold, and you notice this U-rich internal loop. And Nick had circumstantial evidence for the association of the U-rich loop with the poly-A tail. Now, since we know that when RNAs get degraded, the first thing that happens is nucleases chew at the poly-A tail. If you sequester the poly-A tail so those nucleases can't get at it, that would be a very good way of stabilizing the RNA by preventing this first step in decay. And moreover, we had this wonderful example of another non-coding RNA, namely a SNO RNA, and how it interacts with ribosomal RNA in order to introduce a modification of pseudo-U residue at about this position in the ribosomal RNA. And we thought, this is perfect. It's going to look exactly like this. So a new postdoc who came to the lab at that point, Rachel Minton-Fry, decided she was going to do structural studies in order to show that this model was true. She did both NMR and then got crystals of a somewhat truncated E&E complex, not with its own poly-A tail, but with an oligo A9, and did the crystal structure at 2.5 angstroms resolution, along with a lot of help from Tom Steitz's lab. And contrary to what we expected to see this. What you saw is that the E&E binds to the oligo A9 by forming a triple helical structure that you see pictured here. And over here, to remind you, this is what the base triples in a triple helix look like. So there's a standard Watson-Crick UA base pair that's extended by a Hugh-Kupstein pair with an additional U residue. CGC can also form, and note that it's isosteric, a base triple, but in this case, oh boy, you can barely see it there. There's actually a hydrogen here whose presence is pH sensitive, so this particular triple is stabilized depending on the pH. So there's actually supposed to be, in the picture, an H plus right there. So the thing that I found really wonderful about this structure, so this was the longest triple helical stretch that had yet been documented in any naturally occurring RNA. And what these triples look like is exactly what was predicted back in 1957 by Gary Felsenfeld, David Davies, and Alexander Rich, who studied complexes of two strands of poly-U with poly-A. And it's exactly what they predicted based on their diffraction studies of those polymers. So let me say a little bit more about this structure, because this is still sort of introduction. So this isn't what we have. Here are a couple of views of this triple helix. You can see UAU, the triple base pairs. And for people like me who think that are in 2 dimensions, okay, so here are the Watson-Crick base pairs over here, and the Hoofstein pairs. And the triple helix, then the 5 UAUs, is extended by 3 A minor interactions. This is a tertiary structure motif that's found abundantly in ribosomal RNAs now that we have structures of the ribosome. And there are 3 different types here that go deeper and deeper into the minor groove. So let me just okay, summarize. Okay, so what I've shown you, or what I've told you about, is that this is an element that protects the RNA from decay, so we presume it's grabbing the poly-A tail the same way that it grabs oligo-A in the crystal structure, and there are 5 of these UAU triples extended by A minor interactions. And what I didn't tell you, let me get this to behave, is that if you make a mutation of either that U or that U to C, you abolish the stabilization activity. But what Rachel also did was to change both of these to C and then engineer a poly-A tail that had a G there and showed that you could restore the stabilization activity. So that was really good biological evidence that the triple helix is important for the functional RNase resistant. So this raises the question, do other viruses encode pan-RNAs with E and Es? When we started working on this, a lot of virologists, I'm not really a virologist, would say to me, you know, human viruses are different. They sometimes do things that other related viruses don't do. It might be that pan is made only by this human virus and not by related animal viruses. Well, of course, that turns out not to be true. And the second question is, where did the virus get this structure? Are there cellular RNAs that have triple-stranded structures that are used to stabilize those RNAs, and that's what the virus hijacked? So I want to tell you a little bit more about this first, and then go on with unpublished work about the cellular ENEs that we think this came from. So all along we've been trying to find in other RNAs ENE type structures, but it wasn't until we knew about the triple helix that Kazuo Takowski, as a research scientist who's sort of become our lab bioinformaticist, was able to feed into the programs the proper descriptors that allowed us to really search for structure instead of searching for sequence. And when he did that and then searched the viral database, he of course came up with a number of other ENE structures. These ones up here are all in gamma herpes viruses, so things that are somewhat closely related. And one of the fun things is that this particular one was in, so rhesus radenovirus is very closely related to KSHV, and this was in an intergenic region, but in the same part of the genome as PAN was in KSHV. And finding the ENE there meant we could look for the PAN RNA, and when we looked for the PAN RNA, of course, it was there as well. And you notice that all of these have a poly-A addition signal very close by, so that they can make a poly-A tail. And what you also notice is some of these are upside down, which just means that they grab the poly-A tail on the other side of the loop to make the Watson-Crick base pairs, and then use this side for the Hoofstein instead of this way. But either arrangement is quite plausible. So clearly, there are other ones of these. And the conclusions of this work, which were published a while ago, are that they're not that common, but they're in evolutionary diverse viruses. Many herpes viruses have pan-RNAs, presumably having to do something with late protein synthesis. And although they have almost no sequence homology, what they have is a structural homology of this protective element. And what that means is that if we had a good way of searching for structures in RNA, and that's still lacking, we would probably be able to identify lots of other structures, and that's what we're looking for. And recently, Kajikowski has actually looked in a broader database and has found hundreds of ENEs that look like the ones I've just shown you in plant transposons. But in all these cases, the reason there are hundreds is because there are multiple copies of transposons. But one thing that's notable is that the triple helix, the UAUs, are never longer than 6 in a row. And keep that in mind because I'm going to be coming back to that. Okay, so cellular ENEs. Turns out that it was not too hard to identify in 2 very well-studied long non-coding RNAs that are in us and other mammals, the possibility of forming triple helices like this. So this RNA stands for metastasis-associated lung adenocarcinoma transcript, and this stands for multiple endocrine neoplasia. So both these RNAs have been associated with oncogenic phenotypes, although their exact functions are unclear. They're both Pol2 transcripts. They're highly abundant in the nucleus, but exactly what they're doing we don't know. This one is a component of a nuclear body called paraspeckles, but nobody knows what paraspeckles do, so that doesn't really help us with respect to what this RNA is doing. In any case, the interesting thing about both these RNAs is that although they're initially formed with a poly-A signal and a poly-A tail, the major form inside the nucleus of our cells has been processed. And this was discovered by Jeremy Willits, who was a graduate student in David Spector's lab at Cold Spring Harbor. And where their process is RNase P that normally makes the 5' ends of all tRNAs, recognizes this tRNA-like structure, cleaves there or cleaves there, making a shorter form of the RNA. And it turns out that this sequence, this A-rich track with which these RNAs then end, is complementary to the sequences there. So one can predict that they might fold back to form triple helical structures that would look like this. So you notice that these are considerably longer triple helical regions than we saw in the pan-RNA, just the 5 UAUs, but that they're interrupted, so it's sort of a bipartite triple helix by what might be a CGC triple and a CG dinucleotide. And when this was realized, both Jess Brown, who's a postdoc in my lab, and Jeremy Willits, who was in Phil Sharp's lab at MIT until very recently, did mutational studies and just did thermal melting studies that were consistent with the formation of triple helices, as you see here. But of course, what we wanted was the structure to actually prove that these RNAs really do form these triple helical structures. And what I can tell you is that very recently, Jess, with a lot of help from an undergraduate, Kajikowski, a technician, and people in Tom Steitz's lab, David, a graduate student, and Jimmy Wong, research scientist, has very recently actually solved the crystal structure of the mallet-1 ENE and sees this gorgeous triple helical structure that I want to tell you about next. Okay. So here's a little bit for the crystallographers in the crowd. This is all the data I'm going to show you. This is what the electron density map looks like. This is at 3.5 angstroms resolution. And what Jess crystallized was a slightly truncated form of this structure. So she took off the top and added a tetraloop and a little motif that should bind heavy metals for solving the phase problem. She took out a few nucleotides there. And what this over here shows you in this northern plot is that when she inserts this truncated mallet-1 ENE into a reporter RNA that's intronless and polyadenylated, that in fact it stabilizes the core structure, stabilizes to the same extent as the full structure. And in both cases, if you make just one U to C mutation, you lose a lot of the stabilizing influence. So it looked like it was behaving properly. So what have we learned with this new triple helical Here is just the 2D1 and the 3D1, and it's sort of what you might expect. There are two triplex regions that are interrupted at the doublet point. There are, in fact, a minor interactions, but in this case only two. And the triple helix then is flanked by duplex strands of the sort that we saw in the KSHV pan-E&E. So what's going on here? Why does this have this interruption in the middle? One thing that we know, and this is just comparing spatially, just having U triples versus having this interrupted structure, and shows that the distance span is the same, so it's not interfering in that particular way. Base stacking is a very important force in holding a triple helix together, and what's quite clear is that the base stacking that one expects to have with the CGC and the CG is greater. Just look at the overlap in these bases compared to what you would have if you have two U triples next to each other. So that's probably contributing to the stability. What happens at the point of the interruption is that the helical axis gets shifted by 15.4 degrees, so that overall the whole thing turns out to be rather a straight rod, and this may be important for, say, protein binding inside the cell. We don't really know anything about this yet. But the thing that I find most satisfying about it is that there is an explanation that we can get out of this for why we haven't seen, and nobody's reported, triple helices longer than 6 in nature. And it has to do with the fact that there's a potential hydrogen bond formed between the 2' hydroxyl of the boostine strand and one of the backbone phosphates of the Watson strand. And that's illustrated here. This is the bond we're talking about. Here's the backbone, and here's the, I got that right? No, here's the 2', and there's the backbone. There, and it's seen sideways here. And it turns out that the length, you can't really see the hydrogen bond, so this is all potential hydrogen bond. But the distance between the two interacting atoms making the hydrogen bond varies as you start at the bottom of the helix and go up. And that's what's shown over here. So at the very bottom, the length of this bond would be 5.1 angstroms. It gets shorter and shorter